Greetings, class. Have you ever wondered how life avoids poisoning itself? Consider carbonic anhydrase, an enzyme that operates at the incomprehensible speed of removing 600,000 molecules of lethal carbon dioxide per second. Today, we will study enzyme kinetics and immobilization. You know, these tiny molecular machines are literally what keep us alive every single second.
Okay, let's dive right into this. Here is our roadmap for the class. We'll kick things off with an introduction to enzyme speeds, and then move right into understanding maximum rate Vmax. From there, we'll tackle the brilliant mathematical workaround of the double reciprocal plot. After that, it's the Michaelis-Menten constant CAIE, followed by the significance of kinetic values in the real world. And finally, we'll bring it all together by exploring how industry is immobilizing commercial enzymes.
Section 1. Introduction to Enzyme Speeds, the Biological Marvels. So, to really grasp what's happening inside our cells, we've got to understand the sheer mind-boggling speed we're dealing with. First up, we need to define turnover rate. This is simply the number of substrate molecules converted into product by a single enzyme molecule per second. Basically, it's the fundamental metric we use in biochemistry to judge an enzyme's baseline capability. Think of it as the engine's RPM.
These are the key points related to this topic. Focus on them carefully as they are considered essential for your exams.
To put this into perspective, your average enzyme converts about 1,000 substrate molecules per second. Not bad, right? But then you have carbonic anhydrase. This thing is an absolute powerhouse, achieving an immense 600,000 conversions per second. It absolutely has to be that fast to prevent lethal carbon dioxide buildup inside our respiring tissues. We are talking about a reaction occurring 10 to the power of 7 times faster than it would without the enzyme. Absolutely wild. But here's the catch. These extreme speeds rely entirely on rapid cellular diffusion over super short distances. It literally requires tens to hundreds of thousands of rapid molecular collisions per second between the enzyme and the substrate just to hit those numbers. Section 2. Understanding maximum rate Vmax, the measurement problem. Now, knowing that enzymes move at these breakneck speeds, we run into a bit of a scientific mystery. If they're moving this incredibly fast, how do we actually find their theoretical maximum reaction rate? We call this Vmax. At Vmax, all the enzyme's active sites are completely saturated with substrate. To measure this, we keep our enzyme concentration constant and just keep adding more and more substrate, plotting the initial rate against the substrate concentration. But well, this gives us an asymptotic curve. Honestly, an asymptotic curve is kind of like a student's infinite excuses for late homework. We can theoretically reach the end, but in practice, it never completely flattens out. That's because Vmax theoretically occurs at an unplottable infinite substrate concentration. So measuring it directly in a lab, pretty much impossible.
The following are critical points regarding this heading. Please examine them closely as they are highly significant from an examination perspective.
Section 3. The double reciprocal plot, the mathematical solution. If we can't exactly measure infinity, we need a workaround. Enter the elegant mathematical solution devised by scientists.
What's really interesting here is how flipping the data completely solves our problem. Reciprocal, mathematically, means the inverse of a value. So instead of our standard plot, we plot 1 over the substrate concentration on the x-axis and 1 over the velocity on the y-axis. We call this a double reciprocal plot. Why do this? Well, it neatly overcomes the infinity problem, because 1 divided by infinity equals exactly 0. 0 is a number we can actually work with. Just like magic, this simple transformation turns our impossible, never-ending asymptotic curve into a nice, clean, straight-line graph.
So, how does this straight line actually help us calculate Vmax? We take our specimen results to plot the line, and then we strictly look for where it crosses the y-axis. At this exact y-intercept, the value on our x-axis, which is 1 over the substrate concentration, is exactly 0. And since 0 mathematically represents infinite substrate, that y-axis intersection point perfectly equals 1 over Vmax. It's so clever. From there, it's effortless. You just invert that single y-intercept value, and bam, you've calculated your theoretical maximum rate, Vmax.
Here are the core details regarding this topic. Review them intently for they are deemed crucial for your finals.
Section 4. The Michaelis-Menten constant, Km, the efficiency metrics. But Vmax is only half the story. Let's look at the second crucial kinetic value we can pull from our graph. The other crucial value is Km. Km is defined as the exact substrate concentration when the enzyme is operating at exactly half of its maximum rate, so half Vmax. This indicates the point where half of all active sites are occupied. Now, the crucial takeaway here is that Km serves as the definitive measurement of enzyme substrate affinity. Basically, how eagerly does the enzyme grab its target? A higher affinity results in a numerically lower Km value. And we can find this effortlessly on our double reciprocal plot. Look at where the straight line intersects the negative x-axis. That x-axis intersection point gives us the value of negative 1 over Km, allowing us to calculate Km instantly. To really lock this in your memory, let's use a car analogy. Think of Vmax as the absolute top speed of a sports car, the highest possible reaction rate. Km, on the other hand, is your acceleration, the enzyme's affinity, or how quickly it actually gets to that top speed. Both are totally essential for efficiency, but they measure completely different things. And looking at these turnover numbers brilliantly illustrates that exact point. Notice the massive natural variation here. We've got carbonic anhydrase up top humming along at 600,000, and way down at the bottom, lysozyme operating at just half a molecule per second. This great variation proves for sure that top speed, Vmax, and acceleration, Km, are completely independent traits. An enzyme could have a massive top speed but terrible acceleration, or vice versa. Section 5. Significance of kinetic values. Real-world application. Now, you might be wondering, why do we actually care about all this math? Well, these numbers aren't just here to torch you on exams. They literally drive modern commercial biochemistry. Scientists use these practical kinetic values to create incredibly detailed computerized models of biochemical pathways. This helps us predict how entire whole cell environments will behave in the real world. These models can factor in changing conditions like temperature and pH. It allows us to quantitatively compare an enzyme's preference for different substrates, and most importantly, it directly guides genetic engineering so we can design highly efficient, entirely new future catalysts. Section 6. Immobilizing commercial enzymes. Practical magic. Speaking of the commercial world, let's shift to the industrial scale to see how we reuse these expensive biological machines. Commercial enzymes have enormous applications, but they are incredibly expensive. Corporations heavily rely on recycling them to keep their production costs down. A brilliant example of this is lactase. Instead of just dumping it into a vat of milk and losing it forever, lactase is successfully immobilized by trapping it inside tiny alginate beads. Then, liquid milk is continuously run through columns packed with these lactase-containing beads. The enzyme hydrolyzes the milk's lactose into simple glucose and galactose, efficiently producing commercial lactose-free dairy products. So why does this beat just mixing the enzyme directly into the product? First off, it completely avoids losing your costly enzymes during processing. It also ensures the final commercial product remains totally free of enzyme contamination, while enabling continuous, highly profitable enzyme reuse over time. But biophysically, it gets even cooler. This firm alginate embedding structurally restricts the enzyme, which physically prevents molecular denaturation. This provides the enzyme with significantly higher resistance to extreme pH changes and vastly increases its tolerance to severe commercial temperature fluctuations. All right, class, let's do a quick high-energy summary to lock these absolute core concepts in for your exams. Turnover rate defines your baseline catalytic speed. Vmax represents an enzyme's theoretical maximum reaction velocity. Since we can't measure infinite substrate, we know double reciprocal plots solve that limit by finding Vmax perfectly at the y-intercept. The Michaelis-Menten constant, Km, defines affinity and is found right there at the x-intercept. We use these kinetic values for advanced predictive computer modeling. And lastly, industrial alginate immobilization firmly prevents denaturation and allows for continuous recycling. Before we wrap up, I'm going to leave you with this ultimate test of understanding question. If an immobilized enzyme is structurally restricted by the alginate beads, how might this physical restriction impact its Km value compared to when it's free in a solution? Really think deeply about how physical shape and access affect that affinity. Thank you so much for joining this masterclass. Keep asking questions, and I'll see you next time.
Key terms for this topic are presented on the screen, classified according to different exam boards. These terms require dedicated focus during preparations as they are essential and should never be ignored. In addition, past paper analysis is included for each board and paper type. Be sure to review this carefully as it will greatly contribute to improving your preparations. Also, make an effort to understand the meaning and application of each keyword rather than memorizing them on their own. Regular practice using these terms in past paper questions will significantly enhance your accuracy and overall exam performance.
Please subscribe our channel to get more educational videos. Thank you.